In this final section, I want that you identified some of the examples of mutation that we just studied. So I want that you try to identify if there are some nonsense mutation, some missense mutation, or a type of point mutation. So let's start. Now we're going to do an example of a point mutation. Here's your wild type, your DNA template strand. Here it will be your complementary strand, your messenger RNA, your protein. Here you have your initial amino acid, and here your stop codon. So I want that you see what happened here. What happened if you replace an A, you have an A instead of a G. So you're going to change this one, and this one, and you will see what happened here. So we'll stop the video and you can find the answer. So here you can see that as you change an A here, and the complementary will be a T. So this will be a difference in your complementary strand. Here you will have a U because the complementary, well, G is the complementary of C. Now, in the case of A, the complementary will be U in the RNA. And now if you go and if you see if there was a difference, you found that actually there was no difference, because remember the genetic code is redundant. And here you find this is an example of a silent mutation because there was no effect on the amino acid sequence. So in this case, this point mutation didn't have an effect. Here you have another exercise, and you have again all the information here. And here the change is that it was a T is replacing a C. So here all this one will change. So try to find the answer. So please stop the video and try to find the answer. And the answer is the following. The T now will produce an A instead of a G. And here instead of G, you will have an A. And again, here you will have a different one. So instead of glycine, you have serine. And as a result of that, you would have a missense mutation. Now another exercise. Let's see what happened. Again, the same dynamic, but now instead of an A, you have an A instead of a T. So all of this one will change. So please stop the video and try to answer this question. So here you will have a T. Here you will have a U. And here you will get a stop codon. So this will be a nonsense mutation. Now this is another exercise. Here you have an extra A. So I want that you tell me what you will find. Here you will get a T. Here you will have an extra U. And here you will have a stop codon. So there is a frame shift causing an immediate nonsense mutation due to a one base pair insertion. Now let's try to solve this one. Now you are missing an A. And as a result, you're missing here a U. Here you change, and now you have leucine and alanine. And as a result, there was a frame shift causing an extensive missense of one base pair deletion. Now let's go and do this exercise. So now you're missing an entire codon. Just stop the video again and try to answer the question. So this will be your complementary strand. This will be your messenger RNA. Here you will get a stop codon, and there will be no frame shift, but one amino acid is missing due to the 3 base pair deletion.